Bismillah ar-Rahman ar-Rahim. My lecture today is titled Neuroglial Cells. كتعريف بسيط لنيوروجليال cell or glial cells, they are referred to as neuroglial cells or simply glia cells. Don't participate in the receiving and sending electrical impulses. Various types of neuroglial are found in the brain. Those are the cells which support and protect the neuron within the CNS, central neuroglia, or PNS, peripheral neuroglial cells. What are the main functions of the glial cells? There are many functions of the glial cell. 1. Physical support or protection for neurons and insulation for nerve cell bodies and processes. 2. Repair of neural injury and metabolic exchange between the vascular system and the neurons of the nerve system. 3. To destroy pathogens and remove dead neurons. The glial cells are smaller but are more in number, unlike the nerve cell glial cells. 4. Have the ability to divide it. What type of the neuroglial cells can be classified for type of neuroglial cell for two types. In central neuroglial cells, in CNS or central nervous system, and peripheral neuroglial cells. In central neuroglial cells that can be found astrocytes, oligodendrites, microglia, and ependymal cells. In peripheral neuroglial cell can be found two cells, Schwann cell and satellite cells. This figure shows the many types of the neuroglial cells in central nervous system or peripheral nervous system. Can be see the astrocytes in two types, and the microglial cells, that is small cells, many of the processes, and oligodendrites and ependymal cells. But in central nervous system, peripheral nervous system, that can be shown the Schwann cells. Astrocytes, they are large and star in shape, consist of many branched cytoplasmic process, and it contains intermediate filament composed of a distinct protein called glial fibrillary acidic protein. Astrocytes also have prevascular feet to form blood-brain barrier. Astrocytes also provide nutrients to neurons and produce as a hormone known as glial-derived growth factors, which someday might be used as a cure for Parkinson's disease and other disease caused by the neuron degeneration. Also, astrocytes can be seen in many of functions, can be participate in controlling the ionic and chemical environment of a neuron. Astrocytes also bind neurons to capillaries and to the perimeters. Perimeter, that is the thin connective tissue that covers the central nervous system, and to blood-brain barrier. Astrocytes also have ability to convert glucose into glycogen for storage and use when needed to extend neuron power in case of feeling blood sugar level. Astrocytes also play role to tissue repair by processes for called glycolysis about proliferation of astrocyte for full damage area. There are two types of astrocyte in central nervous system. Number one, that is the protoplasmic astrocyte and fibrous astrocyte. Protoplasmic astrocyte that have many of properties that different from the fibrous astrocyte. The protoplasmic astrocyte, these cells have short and thick processes with many branches. They are merely located in the gray matters of brain and spinal cord. The main function is supported neuron in a metabolism. But in fibrous, another type of the astrocyte in central nervous system, these cells have long and thin processes with a few branched. They are mainly located in the white matter of brain and spinal cord. The main functions in this type of astrocyte that binding and supporting the neurons. Oligodendrites. This type of the neuroglial cells in central nervous system. There are type of glial cells in central nervous system that creates myelin sheets around the axon of neurons. The creation of myelin allow for the formation of myelin sheets around the neural axon. This myelin sheet allows for action potential to travel down the axon much more efficiently and quickly compared to unmyelinated axon. And these cells or the single cells of this type can be inside many axons by their cytoplasmic process. And their cells located in both gray and white matters. The another type of neuroglial cells in central nervous system, there are microglia. This type of the cells, they are small elongated cells with short process. Nuclei of the microglial cell can be recognized in routine hematoxylin and eosin preparations by their dense elongated structure. It is originated from bone marrow monocytes, while the other glial cells originated from the neural tube. These cells have capacity for phagocytosis to eliminate the foreign materials. They processes small, round, constant nucleus and only a few short processes. Another type of the central nervous system neuroglial cells, that ependymal cells. This type of the cells, they are low columnar epithelial lining the ventricles of the brain and central canal of the spinal cord. In these cells, in some location, ependymal cells have cilia like structure which facilitated the movement of cerebrospinal fluid CSF. Ependymal form with blood vessels, the choroid plexus that from the CSF. Ependymal cells also have involved with binding between neurons by distal folding process. In peripheral nerve system that's found in the many of the peripheral neuroglial cells that merely found in Schwann cells, that is the principles of glial cells of peripheral neurons or peripheral nerve system. These cells can be found in two types of Schwann cells. That is myelinating and in-myelinating Schwann cell. Myelinating Schwann cells are around axon of motors and sensory neuron to form a myelin sheet. Members or membranes of Schwann cell have a higher proportion of lipids than do other cell membranes, and then myelin sheets serve to protect axon and maintain as a constant ionic microenvironment required for the action potential. Between of adjacent Schwann cells, the myelin sheath show small nodal gap along the axon called node of Renver. The length of axon covered by the one Schwann cell is called the internode segment and may be more than 1 millimeter. The structure of Schwann cells associated with the unmyelinated axon differs from myelinated axon were detected in the organization of the granular endoplasmic reticulum. In the presence of cilia and in the percentage of cytoplasmic occupied by the virus components. What are the main functions of the Schwann cells? There are many main functions of this Schwann cells in peripheral nerve system. One, the conduction of the neuron impulse along axon. Two, nerve development and regeneration, trophic support for neuron. Three, production of the neuron extracellular matrix. Four, modulation of the neuromuscular sympathetic activity. 5. Presentation of antigens to lymphocytes. Another type of the neuroglial cell in peripheral nerve system that is satellite cells. This cell that small cells from a covering layer over the large neural cells bodies and the ganglia. The ganglia that collection of the neuron outside in the CNS. تجمع من الخلايا العصبية تكون خارج السنترال nervous system. The satellite cell exert a trophic or supportive effect on this neuron. Solutions, nourishing, and create suitable microenvironment to the neurons. Thank you for your listening. شكرا جزيلا أحبتي الطلبة. وإن شاء الله تكون المحاضرة مفيدة للجميع. شكرا جزيلا.